NAG is an established biomarker for the detection of renal disease and nephrotoxicity. It can be measured in urine and also it can be measured in serum. NAG kit is FDA approved and CE registered. The kit is available in 3 different sizes. The choice of kit depends on the number of samples to be assayed. Now we can discuss how we can use the NAG kit. So this is an example of the kit. This is the 1290050 kit, and it comes in a box, as you can see, and inside the box are a number of method sheets or information sheets. The first sheet is the manual method, and this, apart from details of how you carry out the procedure, contains information on normal reference ranges for males and females. The second sheet is how you formulate the calibrant, or if you've ordered controls, use the same procedure. The third sheet we've just put in as an example. This is a method sheet for automated system on the Olympus. The actual box contains 2 bottles of liquid. The first one, N, which is the substrate, the yellow liquid. The second liquid is buffer G, which is a stop-in buffer. And the third bottle contains a solid, which is solid buffer A. Final contents of the box are 2 bottles of calibrant, so this shows the entire contents of the kit and all that is required to carry out the manual assay. So how do we prepare the kit for use? Well, the first thing we do is mix buffer N and buffer A, substrate solution N and reaction buffer A. This can be done simply by pouring N into A, so add the liquid to the solid, and it pretty instantly dissolves, but we have to make sure it's 100% dissolved. And this is done by replacing the lid and inverting and shaking. So N plus A is then ready for use. It's stable for 7 days at 2 to 8 degrees, so it can be used at different times during that period. The next thing to do is reconstitute the lyophilized or freeze-dried calibrants. This is done by adding 1 ml of distilled water, and you see it pretty instantly dissolves. But again, we've got to make sure we have 100% in solution. And this we do the same procedure, invert and shake. But then we leave it at room temperature for 20 minutes to make sure that it's all dissolved. Now, actually carrying out the assay procedure is very simple. First thing you need is you do need a control, and usually this is distilled water. So we've bet 50 microliters distilled water. A similar amount, 50 microliters, of the calibrant pipetted. And as we require a particular accurate answer or value for this material, we usually have duplicates or more just to make absolutely certain. Similarly, the 50 microliters of the sample is pipetted. So now it comes to add in N plus A, and in this case we add 750 microliters, and this is added to the control, the calibrant and the sample. The samples and calibrant and controls are now incubated at 37 degrees and water bath for 30 minutes. Once that is over, we then need to add stopping buffer G. In this case we add 250 microliters. Once you add the stopping buffer to the calibrant, you get an almost immediate intense red colour. After 10 minutes we need to read this, so the contents of each tube is transferred to an individual vial, which can be read in the spectrophotometer. So any instrument that reads at 505 nanometers or near to it can be used to carry out this assay, so it's very adaptable to almost any type of laboratory. Once we have the values, then the actual NAG activity in the urine is calculated using the equation given in the method sheet, incorporating the calibrant. So now we come to the semi-automated procedure, and there's an issued method sheet with this. So again we have a control, which in this case is distilled water, and we put 16 microliters into the 96 well plate. So now we add 16 microliters of calibrant. This is reconstituted as we saw earlier. So now we add the sample. Again we use 16 microliters. The point you've probably realised is that it's a much smaller volume, so it's much more economical. It's particularly useful if for example, in an animal study where you have low volumes of urine, or you need to preserve the volume of sample. This is 16 microliters compared with 50 microliters in the manual method. You can add controls. We have high, medium, low controls, and these are very useful, certainly at the beginning of a study or a trial, to actually look at, control the range of the assay, particularly if you initially don't know the level of activity of the enzyme that might be excreted or you might find. The next stage is adding 240 microliters of solution N plus A. As you realise, this is smaller volume, so you can get a greater number of assays from the same kit. using this procedure. Now, because of the smaller volumes, you have to be careful mixing, so we normally shake for 5 to 7 minutes. This type of shaker is usually available in most laboratories. Put the plate into an air-heated incubator, which is maintained at 37 degrees, and this is usually for 30 minutes. When the 30 minutes is over, we then add 18 microliters of stopping buffer G. The advantage of this procedure, apart from the lower volume and saving the reagent, given the potential for a greater number of assays, is the use of a multiple PET, which makes analysis very easy to carry out. You notice that the colour is there, but in this case it's less intense. That's because of the amount of material present is much lower. So now we move to the 96 well reader or ELISA plate reader. You can set the wavelength anywhere between 490 and 520 is okay, but the nearer you can get to 505 the better. And the advantage of the semi-automated methods are the smaller volumes used enable a larger number of samples to be assayed. Reading is automated and instantaneous. Most laboratories have the equipment required. So now we move to the automated procedure. The first thing is to add to the containers which are provided or specific for the particular machine. So they're shaped. We then pour stopping buffer G into a similar container. So that's essentially similar to the other two procedures, except we now have them in dedicated containers for the particular instrument. So the separate reagent containers are placed into the reagent chamber. So that's now the reagent tray. You note the position where they are in the tray. The reagent tray is refrigerated at 4 degrees, so you can keep things there overnight, for example. The calibrant, and these go into 3 ml plastic cups. And in this case they're loaded into the sample tray. This is not refrigerated. So for every assay you would need new samples. We then set the required parameters using the computer. So the calibrating QC is now okay. At that point we can add the sample again into 3 ml plastic vials. And again, the position in the tray is noted on the computer, because it may be in some situations there'll be different assays running at the same time. So we set the parameters. Now you can start the assays, and they're transferred into a 37-degree water bath. The NAG kit is available in 3 different forms. The 1290050 is capable of assaying 300 tests in an automated system and 50 tests if used manually. This is the most popular test kit of the 3. 1290-100 can carry out 600 tests in an automated machine and 100 tests if used manually. 1085 is rather different. It's designed for smaller batch assays and contains buffer A divided into 5 bottles, so that assays can be carried out over a period of 10 days. The sensitivity of the kit ranges from 0.25 international units per litre to 200 international units per litre. Please visit our website for more information.